Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So thank you so much for joining my new beginning today. So today we're going to talk about double fertilization. For all those who are new, Monday, Wednesday, Friday we talk about a class 12 chapter. Tuesday, Thursday, Saturday we talk about a class 11 chapter. So today is Monday, so I hope you guys had a great weekend. So today we're talking about a class 12 chapter, that is double fertilization. So we are done with the first three parts of this chapter where we spoke about the male gamete, how the male gamete is formed, how the female gamete is formed, how the process of pollination happens. So today, once after pollination happens, it's going to be double fertilization. So today we are going to talk about how the process of double fertilization happens. So once we talk about double fertilization, so then we are almost towards the end of this chapter. So we have the next topic will be the development of seed, the fruit, etc. So we collectively call that as the post-fertilization development. So that will be on Wednesday. So by Wednesday we'll wind off this chapter, so we can say goodbye to the first chapter in class 12. So well, in class 12 there's also another chapter called as reproduction in organism. Then you have sexual reproduction in flowering plant. So reproduction in organism I'm not teaching because for the last 2 years there's no question in this chapter, and it's pretty simple also. So once I teach you human reproduction, sexual reproduction, biological classification, plant kingdom, so things become very clear in that chapter. So at the end we will revise that chapter. All right, so yeah, so this week you have 2 Menti quiz, so I would like to give you the update on the same. So let's move forward. Yes, this is the update that I want to give you. So we have a Menti quiz for all those who were asking me for the Menti quiz. So when is the Menti quiz? So the Menti quiz is here. So the Menti quiz is on 2nd December, that is Thursday. So on Thursday at 5:15. So you have the Menti quiz. So please do join the Menti quiz. So thank you DJ Films and Entertainment for joining us today. So please do like, share and subscribe. Yes, and thank you for your support. Right. So please do join us on 2nd of December, so at exactly 5:15. So if you guys have an issue with 5:15, so we can make it 5 or exactly 5:30. But 5:15 would do because by the time you guys join, it would be 5:30, and then 5:30 we could begin the lecture. Sorry, we could begin the Menti quiz. So the topic is neural control and coordination. So I have taught neural control and coordination under the New Beginning series. So I wish I want you guys to join for this Menti, because it's going to be a very easy chapter. So we're just going to play for 25 questions. So please do join. So please share, tell your friends to join. So please share the link with your friends. So one status update will help us do the needful, right? So please do all that, and I wish to see you on 2nd of December, and I also wish to know who is the winner. So the more the number of people, more the fun. So please do join me for the same. Well, so in the previous lecture, that is on Friday, we were talking about the process of pollination. So how pollination happens, what are the 3 types of pollination, so what are the agents for pollination, etc. So quickly I'll give you a recap of all that, and then we'll move to the process of double fertilization, that is today's topic. So pollination is when your male gamete lands on the pigma. Sorry, when your male gamete, that is the pollen grain, it lands on your stigma after dehiscence from the anther. So that is your process of pollination, right? So it is a step before fertilization. So after gametogenesis, the next step is pollination, because once the gametes are formed, the gametes should meet. So how are the gametes going to meet? So that is by the process of pollination, and once pollination happens, double fertilization will proceed with. So pollination, so we spoke about 3 types of pollination. So the first type of pollination is your autogamy. So the second one is your geitonogamy. The third one is your xenogamy. So these are the 3 types of pollination. So what is autogamy? So auto is self. So when pollination happens within the same flower. So that is autogamy, right? So now what is geitonogamy? Where it happens within the same plant which has 2 flowers. This is within the same plant which has 2 flowers. One is the male flower, one is the female flower. So xenogamy, so this happens between 2 different plants. So this is basically or typically your cross pollination. So these are the types of pollinating agents. right? So when you talk about autogamy, that is self-pollination basically. So autogamy can have two types of flowers. So what are the two types of flowers in autogamy? So autogamy can have cleistogamous or chasmogamous flower. So it is chasmogamous or cleistogamous. So chasmogamous is when the flower is well open, it's well exposed, the anther and the stigma are well exposed, so that is your chasmogamous. So cleistogamous is when the flower is in the bud stage, when the male and the female are within the same flower, they are very close, so they do not open at all. So here invariably your autogamy happens. So autogamy ensures an assured seed set even without the agents of pollination. So with this we concluded your autogamy. So then we went on to talk about inbreeding, outbreeding techniques. So what happens when you have continuous inbreeding? So also we spoke about your pollinating agents. I am coming to the pollinating agents. Yes ma'am, a very happy good evening. So thank you so much for joining our lecture today. So please do like, share and subscribe. Hello Sneha, Sunil, hello, a very happy good evening. So thank you so much for joining my lecture today. So I guess you're new to my lecture, so thank you so much for joining us today. So I am talking about the process of pollination. So the lecture on pollination is already over. I am just giving you a recap. So before we go into agents of pollination, so what happens when a plant continuously undergoes self-pollination? So when a plant continuously undergoes self-pollination, it leads to inbreeding depression. So this inbreeding depression leads to reduced fertility. Right? So the plants naturally go for cross-pollination and prevent your self-pollination. So how does the plant do? So first thing is they ensure self-incompatibility. They ensure that the pollen grains on the stigma does not undergo fertilization. So then you have this immature or pre-development. So either the anther develops first or the stigma develops first. So when the anther develops first, that is proto-andry. When the gynoecium matures first, that is proto-gyne. Also maybe the stamen and the stigma, they can be of different heights, so that pollination is not possible. So that condition is known as heterostylum. And the third one is when they produce unisexual flowers instead of bisexual flowers. So these are the ways by which your inbreeding depression is overcome. So that is your outbreeding devices. So the next thing that we went on to talk about is the agents of pollination. So we have two agents, one is biotic agent, the other one is abiotic agent. So biotic agent means the living organism, so this includes your animals, or you can say insects. So abiotic includes your pollination by wind or by water. So among pollination by wind, so pollination by wind is called as anemophily. So this pollination by wind is more common compared to pollination by water. Okay, so how does wind pollination happen and what are the criteria for wind pollinating pollen to be? So when the pollen grain is pollinated by the wind, the pollen grain should essentially be of light in weight so that it can be easily carried by the dispersed winds. And also the stigma here should be very receptive. It should be very wide open so that it can catch up the flowing pollen grain. So these are some of the characteristics. So here they should have a clustered inflorescence and that ensures your wind pollination. So example will be mostly grasses and your corn tassel or your cob corn. So these are examples of wind pollination. So talking about your wheat is also wind pollinated. So then your water pollination. So water pollination is a bit less rare and only 30 genera of your monocots undergo water pollination. So here we also spoke about one contradiction, that all aquatic plants do not undergo water pollination. So the examples were water lily and hydrilla, right? So they do not undergo water pollination just because they are aquatic plants. So they undergo pollination by the process of anemophily or by animals, right? Okay, so how does water pollination actually occur? So it happens in two ways. When the female flower is on the surface, the male gametes travel to the surface and then pollination happens. When the female flower itself is submerged under the water, so that is the fertilization, the pollination happens inside water. Example here, Zostria. Right, so for all the water pollinated pollen grain, it is surrounded by a mucilaginous sheath to prevent it from getting wet. Right, so that was about the abiotic agents. Now talking about the biotic agents. The biotic agents have a variety. So you have the insects, you have the bees, etc. So bees are the most common type of pollinators, and if you have to if you need an animal or an insect to pollinate, so the flowers should be very colorful and bright so that the insect is attracted. So once the animal or the insect comes to the flower should offer a reward. So if you're going to do a help for me, I have to pay you, or I have to do something in return for you, right? So by keeping the flowers or the petals very bright, so the plant or the flower attracts the insect. So once the insect comes near, the flower should be able to offer something to the insect in the form of nectar or in the form of a safe place to lay its egg. So only then the insect will come. So the moment insect comes, the pollen grains of this animal pollinated plants are very sticky. They stick to the surface of the insect and then they are easily pollinated. So we also spoke about amorphous flowers, and then you have another example of yucca and a moth, right? So these are some examples of your insect pollinated flowers. Right, so sometimes your primates, arboreals, they are also examples of your biotic agents. So with this, we are done with the agents of pollination. So then we had to talk about outbreeding services. So what does outbreeding services mean? So today we'll talk about that. We'll start from there and finish off double fertilization. So artificial hybridization, sometimes you would have seen hibiscus in a different color which does not exist in nature. So sometimes you would have seen a hibiscus that is pink in color. So normally we have the white and the white and red, say for example, but suddenly you get an in-between color, so you get a different color. Say for example, you would have seen cabbage in a different color, that is purple color, right? So these are all hybrids. So how does the hybrid comes? So for crop improvement program, to improve the quality of the crop, to improve the yield of the crop, so sometimes you have to do hybridization, right? So in that context, for crop improvement program, we ensure crossbreeding happens. So in that case, naturally you have to stop the self-fertilization that is happening. So if normally these two plants undergo self-pollination, but for the purpose of your crop improvement program, if I have to stop this self-pollination and ensure cross-pollination happening, so I have to make sure I do some work before. Say for example, I have a flower which is bisexual. It has both male and female. So Sneha says yes. So you have seen the purple cabbage, right? So suppose, imagine I have a flower which is bisexual. It has both the male and the female portion. But for my crop breeding program, I have to ensure only cross pollination happens. Right, so now this plant is bisexual, so I have to prevent it from self-pollinating. So I pick out the anthers away from it. So in that bisexual flowers, I pick out the anther and throw it off so that no self-pollination occurs. So in a bisexual flower, when I remove the anther, so that process is called as emasculation. Emasculating, I am removing the anther. I am removing the masculine portion. Right, so now once I remove the anther or the female portion, now I have the female portion which has the pistil or the carpel, the style stigma and the ovary. Now I cover the style stigma ovary with a butter paper in order to prevent any contamination that can happen later, because I want it pollinated only with the plant that I have decided, right? So by naturally also no other means of pollination should happen, so I cover it with a butter paper. So this is called as bagging technique. So all I want you to do is remember these two terms. So what is emasculation and what is bagging? So when I have a bisexual flower, and from the bisexual flower, if I remove the anthers using the anthers from the flower bud using the forceps, so that is called as emasculation. So once the male portion is removed, the female part is covered with a butter paper, that is bagging. So this is how I emasculate it and this is how I bag it. So this is your outbreeding technique. Alright, so let's move forward to the most awaited topic, that is double fertilization. What is this double fertilization? So first our gametes were ready. So you had your male gamete. So the male gamete is called as the pollen grain. So the pollen grain can either be in the 2-cell stage. So here the 2-cell stage includes the generative cell plus the vegetative cell. Right? So if the pollen grain is in the 3-cell stage, it can be the vegetative cell plus 2 male gametes, right? So now I have the female gamete. The female gamete is the embryo sac. So I hope you guys remember the structure of the embryo sac. The embryo sac is the female gametophyte. So this embryo sac is 7-celled and 8-nucleate. So here if I have 3 cells, this end is called as chalacal end. And these are the antipodal cells. Fine. So in the center I have 2 nuclei. So this is the secondary polar nuclei. Then here I have 3 cells. And here I also have a finger-like apparatus. So this end is known as the micropyle end. This is the egg. And these two are the synergids. So the synergids help in the formation of this finger-like apparatus. So this finger-like apparatus is called as the filiform apparatus. Right? So this entire structure on this side is known as the egg apparatus. So 1, 2, 3, 4, 5, 6, 7, 8. So 8 nucleate. So how many cells are there? 1, 2, 3, 4, 5, 6. The entire one, 7. So this is your embryo sac. Fine. So the male and the female portions are ready. So now pollination has happened. So when pollination has happened, so it means the pollen grain has landed on the stigma. So this is our stigma portion. So this is the style portion. This is the ovary. So inside the ovary, the cavity of the ovary is called as the ovarian cavity. Or the locule. Right? So inside it I have the structure. Yes, Neha, you're right. Now, once the pollen grain has landed here, if it's the right pollen, then the stigma will become compactable. The stigma accepts the pollen and it is now ready for fertilization. Once the stigma recognizes it as the right pollen, so if this pollen is in the 2-cell stage, it will undergo the 3-cell stage. So this generative cell will undergo mitotic division and then it will become the 3-cell stage. So once the pollen grain lands on the stigma, and if it is in the 2-cell stage, it will become the 3-cell stage. Fine. So once it becomes the 3-cell stage, or if it's already in the 3-cell stage, so your pollen grain will germinate on the stigma. So how does it germinate on the stigma? So your pollen grain has 2 layers, right? So the outer layer is called as exine and the inner layer is called as intine. So the outer layer exine is made up of a very tough substance, that is sporopollenin, right? But on the exine layer itself, you have certain structures that is known as germ pores, where your sporopollenin is absent. So these germ spores help in the formation of your pollen tube. So if this is your germ pore, it forms a tube like this, because from here the pollen grain has to reach the embryo sac, right? So it forms a tube like this. So this is the pollen tube. So once the pollen tube is formed, the male gametes enter via the micropyle end. They can also enter via the chelysal end or via the micropyle end. But majority of the plants, the pollen tube enters via the micropyle end. So this is how they enter. So now we have two male gametes with us, right? And it is entering the micropyle end. So it will move via the filiform apparatus and reach the synergids. So it will reach the cytoplasm of the synergid. Now it has two male gametes, right? So one male gamete will fuse with the egg. So when it fuses with the egg, so it leads to the formation of zygote. So this is normal fertilization. So this is known as syngamy. So gamy is fertilization. So syngamy is fusion of the male and the female gamete. But you also have another male gamete, right? So this another male gamete travels up and reaches the secondary polar nuclei. So the other male gamete fuses with the secondary polar nuclei. So when it fuses with the secondary polar nuclei, it forms the primary endosperm nucleus. So see, here you have one male gamete, that is 1N. So here you have two polar nucleus. So two nucleus are there. This is 3N. So now when these two fuses, it becomes 3N. So this is basically triple fusion. So triple fusion leads to the formation of primary endosperm nucleus. Syngamy leads to the formation of your zygote. So since two types of fertilization are happening inside the embryo sac, one after the other or simultaneously, this process is called as double fertilization. So once again, one male gamete fuses with the egg and it forms your zygote. So this fusion is called as syngamy, right? The other male gamete fuses with the secondary polar nuclei and it forms your primary endosperm nucleus, which is 3N. So this is triple fusion. Since there are two fusions happening at the same time, so this process is called as double fertilization. So you understood what is double fertilization? Yes. Alright, so I hope I have made it clear. So now I will show you the picture of double fertilization. So this is how it happens. So you see, so this is the pollen tube. So it is moving through the micropyle end. So towards the filiform apparatus, it reaches. So it goes here. So it fuses with the egg cell, it forms zygote. It fuses with the secondary polar nucleus, forming the endosperm, right? So now after this two fertilization that happens, that is double fertilization. So now here you had antipodal cells in the chelysal end. So the antipodal cells will degenerate. So the synergids will also degenerate. So here you have the zygote. So this is the zygote. And here you have three nucleus. So this is your primary endosperm nucleus. Now this entire cell is no longer called as the embryo sac, it is called as the primary endosperm cell. So the primary endosperm cell has the primary endosperm nucleus and the zygote. So this develops into the endosperm and this develops into the embryo. Clear? So after for double fertilization, your synergids and your antipodals, they degenerate. They are no longer needed. They are just needed for the efficient process of fertilization. So now this will become the zygote. The zygote will develop into the embryo. So this primary endosperm nucleus is now developing into the endosperm, and this entire structure is now called as the primary endosperm cell. Fine? Okay. Yeah, so you can see I have overwritten, so I can rub what I've written. so that it's easy for you to understand from the figure. So like I told you, the synergids degenerate. So you can see the synergids are degenerating. So this is the zygote. So they have changed the structure. Now this is our microphile end. This is the chalacial end. So the antipodal cells are also degenerating. The synergids are also degenerating. So this is my zygote. So this is my primary endosperm nucleus. And this entire structure is my primary endosperm cell. Alright, so this is how the structure is after the process of fertilization. So now moving forward, so whatever I have told you, so that is written in the form of theory, so you can read it when you get my PPT. For all those you are new, you get my PPT from eCareerPoint app. So if you download eCareerPoint app, so you have to log in with your mail ID. So once you log in, so you have a panel on the downside. So there you have a column known as the free stuff. So if you open free stuff, so please select our YouTube channel. So our YouTube channel is eCareerPoint-English. So please go inside. So you have my biology classes. So this series I have named it as New Beginning. So under the New Beginning series, you have all the PPTs uploaded chapter wise. So you can find my PPTs in the form of PDFs there. Okay. Right. So now post-fertilization structure and events. So now when the gametes were ready, so we went to talk about pollination. So pollination is to ensure that both the gametes mate, and once pollination happens, so we also spoke about how fertilization proceeds. Now once fertilization is done, now we're going to talk about post-fertilization development, because after fertilization only you have the formation of the fruit and the seed, right? Yes. So double fertilization followed by events of endosperm and embryo development till the maturation of ovule into seeds and into ovary into fruits. So this process is your post-fertilization events. Okay, so now that we are done with fertilization, now the endosperm will develop, then the zygote will develop into embryo. Now it will form the fruit and the seed. So all these events come under post-fertilization events, the process that happens after fertilization. Alright, so now this is what we are gonna see. So today we're only gonna talk about endosperm development, and then we'll end the lecture. So how zygote happens, or how zygote develops, how the seed is formed, how the fruit is formed, we'll continue in the next lecture. Alright, yeah. So endosperm development precedes the embryo development. So we have both the endosperm and the embryo inside the primary endosperm cell. But now the embryo, the zygote does not develop, first the endosperm develops, right? Because first you have to ensure if your zygote develops that you can provide food for it. Yes, so we always make sure in advance, so if this situation comes, I should be prepared, right? So now the plant is also having the same situation. If the zygote develops, I have to provide the zygote with food. So who will provide the zygote with food? That is your endosperm. So that is the primary reason why your endosperm develops before the zygote, right? So the primary endosperm divides repeatedly, that is triploid, so it forms the endosperm tissue. When a cell repeatedly divides, it forms a group of cells and that becomes a tissue. So this primary endosperm, which is already triploid, undergoes mitosis. So mitosis is equational division, so it is just to increase its population. It becomes a primary endosperm tissue. The cells of the tissue are filled with reserve food material which can support the growing embryo. Right, so most common type of endosperm development is the successive nuclear divisions, and then it becomes a cell. So how this endosperm tissue is formed is by a group of cells, right? So initially I have the primary endosperm nucleus, so that undergoes repeated mitotic division, and only the nucleus divides, the cell wall is not formed. So it is free nuclear. After some time it forms the cell wall, right? Yeah, so one good example of this is your coconut water. So your tender coconut has water, so that is your free nuclear endosperm, and the white color is your primary endosperm cell, right? Yes. So coconut water, tender coconut is your free nuclear endosperm. It is made up of thousands of nucleus. That is by the process of mitotic division. So surrounding the white kernel, so the water is stored inside the white structure, right? So that white structure is your cellular endosperm. Now this is going to nourish the embryo. Okay, so the endosperm may be completely consumed by the developing embryo before the seed maturation. So you need to understand the zygote is going to develop into the embryo is going to become become the seed, right? So when the seed ripens, it's going to become the fruit. Now sometimes so basically we need the endosperm to provide food to the zygote, right? So from the zygote stage till it reaches the seed stage, your endosperm is needed to provide food. Sometimes this endosperm is completely consumed even before the seed matures. Okay, so sometimes it persists even after seed maturation. So there are two types of ways by which a plant consumes the endosperm. So when the endosperm is completely consumed by the plant even before the seed matures, so that is called as endospermous, non-endospermous seeds or non-endospermous plants. So example is peas, groundnut and beans. Okay, so you can remember basically all the dicots, so they have consumed the endosperm completely. So Prince Kumar, hello, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. Fine, so that is completely consumed. Sometimes it persists in the young seed or in the mature seed when it is not completely consumed. So this is called as endospermous seed. This is non-endospermous because endosperm is completely consumed. So this is endospermous because endosperm is still present. So example is your castor and your coconut. Fine. So now let's talk about embryo, just one or two lines. So embryo develops at the micropyle end of the embryo sac because it is towards that end that you have the egg apparatus which has the egg cell. So your embryo development happens at the micropyle end of the embryo sac. So that becomes the zygote. Okay. So most of the zygote divides or begins to divide only after your certain amount of endosperm is formed because it needs energy, right? So you will not run a factory without electricity supply. You will first ensure that there is electricity supply and then go forward recruiting people, right? So much in the same way, so your zygote starts dividing only after certain amount of endosperm is formed. So adaptation, why this happens is because it ensures, okay, you want me to speak slowly? Okay. Sometimes why does this happen? So your zygote begins to divide. So your zygote begins to become an embryo by repeated division. So why does it do so? It does so when your endosperm is done dividing or when your endosperm is half formed. So it does so to ensure that it has enough nutrient supply so that it can grow without any problem. So that is the reason first your endosperm develops and then your zygote develops. Alright. So embryogeny is the early stages of embryo development. So that is a term that we use. So embryogeny is very similar in your monocots and your dicots. So monocots have one cotyledon, dicots have two cotyledon. So cotyledon is a structure inside your seed. So zygote first develops into your pro-embryo, then it becomes globular, that is a ball-like structure, then it has a heart shape and it matures into a well-developed embryo. So first stage is the zygote, the second stage is the embryo, alright? Yeah. So the dicot embryo you can see, so it has two dicots, right? So do I have this figure? Okay, so I will give you the figure of the dicot embryo. So the dicot embryo is a structure like this. You have one cotyledon and you have another cotyledon. And here you have a main axis. So this is the embryonal axis. So the portion of the axis above the cotyledon, above the level of cotyledon is called as epicotyle. So yeah. So the portion, the embryonal axis above the cotyledons is called as epicotyle. So epicotyle gives rise to plumule and your plumule develops into the shoot. So the portion of the embryo axis below the cotyledon, this is called as hypocotyle. And this develops into radicle. So radicle develops into root. Right, so the tip of the root is covered by root hair. So this is the structure of the dicot embryo. So now we are going to talk about the structure of the monocot embryo. So you have a dicot structure, you have a monocot structure. So in a monocot you have only one cotyledon and the cotyledon is here. right? So here the endosperm, can you see the endosperm is here and the cotyledon is here. That is, the embryo is here, the endosperm is here. So you have an in-between layer that is called as the alluron layer. It is a protein-rich layer. So this layer is your alluron layer which separates them, right? So your endosperm, so your cotyledon here is surrounded by a sheath that is called as scutellum. So scutellum is the sheath that surrounds the monocot embryo, right? So now here you have your plumule and your radicle. The portion that gives rise to your shoot is your plumule. So that plumule is again surrounded by a covering that is known as cleoptile. So cleoptile covers your plumule, and then your radicle which gives rise to root that is covered by your cleorhiza. So this is the structure of your monocot seed, right? I hope it's clear. So once again, for all those who have not understood, so let me find a blank slide to explain it to you. So I guess it's pretty clear. So I can explain it here. So monocot and dicot. So in dicot you have a structure like this. So you have your endosperm. So this is the endosperm. So here you have a layer that is your alluron layer. So here it is your embryo. So here there is only one cotyledon. So that one cotyledon is surrounded by a sheath. So that sheath is called as scutellum. So here also you have the radicle and here you have the plumule. So the radicle also has a covering that is known as cleorhiza. So the plumule has a covering that is known as cleoptile. Fine. So this is the structure of your monocot embryo. So we are almost done with one part. So we spoke about how the endosperm develops and how the embryo also develops. So what is remaining is we're going to talk about fruit and seed in the next class, and we'll be almost done. So this I have explained. So we'll be almost done with this chapter. Right, so we are done with the post-fertilization events till your endosperm development and your zygote development to embryo. So what is remaining is the structure of your seed and the types of fruits and how a seed dispersal happens. What is polyembryony? What is apomixis? So we'll be done with this chapter on Wednesday. So today this is the last topic and this is my last slide. So once again a gentle reminder. So we are going to play a Menti quiz on 2nd of December 5:15 p.m. on the topic neural control and coordination. So please join me on the same. So we are going to play for 25 questions. Please do join me. So the winner will have exciting prizes. So please make sure you join this Menti quiz on 2nd of December, that is Thursday. Right, so I have taught neural control and coordination in the new beginning series. You can watch my lecture and then you can come back to me if you have any doubts, and then we'll go ahead with the Menti. So meanwhile we'll finish this chapter on Wednesday, but tomorrow at 5:15 p.m. we are talking about a class 11 topic. So we are currently dealing with plant physiology, so we'll be talking about the last bit of your transport in plants. So tomorrow we'll probably be done with transport in plants as well. So please make sure you join me for the same tomorrow as well. So if you genuinely like my way of teaching, so please do like, share and subscribe. So thank you so much for watching my lecture, and don't forget to lend your support to us. So please spread the word. So this is me signing off. So until tomorrow 5:15 p.m. So thank you and take care.